let us move on to the next topic, which is going to be the major focal point of discussion today, and that is regarding the application of root mean square deviation in deciding if the system has converged when you are performing molecular dynamics simulation. Okay, so now everything must be appearing to you that everything is alien. Okay, so let me explain you everything in quite bit more details so that you can grasp कि यहाँ पर क्या होने वाला, right? So now, have you heard the term this molecular dynamics?
Yes, but not very detailed.
Okay, okay. So, can you just let me know what are your opinions or ideas about what molecular dynamics is all about? नहीं? Okay, ठीक है, कोई दिक्कत नहीं. Uh, you know, to begin with, I would like to uh, extend our concept of protein folding and our understanding of protein structures in a way that whatever properties of the protein that we have learned so far, it all appeared to be static, right? The protein appears to be static in each and every model that we have studied so far. बरोबर है ना? But the thing is, at physiological condition, proteins do not remain static. Okay, they involve dynamics. That means there are continuous motions that are always going on in your protein structure. Okay, and these motions somehow give your protein some functional status, right? So there might be motion in your protein that would enable it to rearrange its secondary structures, okay, in such a way that it can attain an open state or closed state, so that it can attain active conformation from inactive conformation. So all of these things usually happen when protein move. Some small portion of your protein might move, some loops might move, or even protein might undergo global conformational changes. Well, say for example, in case of allosteric protein, there are global conformational changes that might expose certain sites when a ligand goes and attaches to it or in some another way. So all in all, what I want to convey from this discussion is that the proteins are not static. Well, they undergo a lot of conformational changes. Okay, the proteins are dynamic, and owing to its dynamic nature, the protein can undergo a lot of conformational changes, and these conformational changes sometimes give them the functional activity or inactivity that would determine its functionally active state or inactive state, and so on and so forth. Okay, so now the thing is that you can model these dynamics. Okay, these conformational changes can somehow be modeled if you have the initial structure of your protein available. ओके, ये जो मोशन होता है आपके प्रोटीन में, आप वो मोशन को मॉडल कर सकते हैं. Okay, so and to perform such a modeling task of modeling the motion that is involved in your protein, the technique that you use to do this task is called as molecular dynamics. Okay, so this molecular dynamics is a technique which is generally used to model the motion that is involved in your protein. Okay, and we say that you perform such molecular dynamics simulations. You would simulate how the motion takes place in your protein. how the atom would move with respect to each other when a protein is present in, say, some aqueous state, or we'll say for example in your in vivo condition, right? So the technique that is used to model such movement is called as molecular dynamics and simulation. Okay, is it clear now?
Yes, sir.
Okay, so if you want to simulate the motion of your protein, you would be doing this molecular dynamics and simulation. Okay, is it clear now? Okay, now
Yes.
let me show you a typical result of molecular dynamics simulation, right? So that you would clearly appreciate how things happen in such molecular dynamics simulation. Okay, so in order to perform molecular dynamics simulation, well, there are a lot of software. Okay, so well, one of the most popular software that is used for performing molecular dynamics simulation is NAMD, or sometimes people also refer it to as NAMD. Okay, so this NAMD or NAMD is a popular molecular dynamics package that is used to perform molecular dynamic simulation of your protein and protein ligand systems if you want to do it on Windows operating system. Okay, so most of you must be using Windows operating system. So generally, if you are working on Windows operating system, people normally use this NAMD. Okay, there is another package called as GROMAX. This GROMAX is also used for performing MD simulation, but this GROMAX is usually implemented on Linux based system. Okay, so if you are working on non Windows system, well, say for example like Ubuntu or Red Hat Linux, then you would be using this GROMAX package. Okay, so since we are working on Windows, whatever I am going to show you, well, that has those simulation were carried out using NAMD. Okay, and once you carry out those simulation, you can visualize the simulation using the program called as VMD that you are currently seeing. Okay, सबको दिख रहा है ये? Is my
Yes, sir.
screen visible to everyone?
Yes, yes,
So
yes.
whatever is present on screen now is a software which is used for visualizing those simulation. Okay, so this VMD is a program that is used for visualization of the simulation that you have performed using NAMD or NAMD. Okay, so well, first let me load the result of the simulation first, and I would tell you ki bhai, what those simulations are all about. Okay, so whenever you load this VMD, okay, let it be, we don't need that. So what you need to do is that you need to load the PSF file first. Okay, so I will just let you know how things are done over here. Okay, this is called as PSF file, as you can see over here. Open and then load, right? And then what I would be doing is that I would browse and again go to the same location and load a trajectory file. So I am looking out for the trajectory file. This is called as trajectory file. A file name with extension DCD, it is called as trajectory file. Okay, now what is happening over here? Okay, so now you see that this is a water box, okay, in which I have simulated the motion of a protein called as cyclooxygenase 2 that is present in complex with its inhibitor diosmin. Okay, so whatever you are visualizing now is that we have simulated this protein inhibitor complex. Okay, the protein is COX2 and the inhibitor that we have tested is diosmin. Okay, so now in what do we do in molecular dynamics simulation is that we try to simulate the trajectory, or we try to simulate the behavior of this protein which is placed in this box, water box. You can see that the protein has somehow moved out of this water box. It might appear like that, right? So initially the protein was placed in this water box, but in due course of simulation you might see कि ये protein water box छोड़कर भाग गया. Well, but that is not the case. Why? Because we have implemented something called as periodic boundary condition. Well, let us not go into more details of that. Okay, so the thing is that it is only the matter of representation that it appears that the protein has moved out of the box. Okay, but after implementing periodic boundary condition, that will not remain the case. I would explain you everything, right? So what we have done is that you can see over here that there are 1,000 frames. Okay, this is 0th frame, then second, fourth, the number of frames now increase over here. So what we have done is that we have simulated this protein under this aqueous condition. Okay, and after simulating, this is, you say, the trajectory that is obtained for 50 nanosecond. तो ये simulation अपन ने कितने time के लिए किया था? We have simulated this protein ligand complex for 50 nanosecond. Okay, and in this simulation of 50 nanosecond, I have tried to take a you can say I have tried to save the state of this system after every 500 picosecond. Okay, so do you get this thing? Ki what first I have done, I have simulated this protein ligand complex, that is my COX2 diosmin complex. Okay, for how much time? Simulations are done for say some finite times. You cannot simulate for say 1 hour, right? It would be very that is not practical so far the computational tools are available. Okay, so this is a 50 nanosecond simulation. Okay, and in this 50 nanosecond simulation, what I have done is that I have tried to save the state of my system after every 500 picosecond. So whatever frame do you see over here, these are 1,000 frames. That would mean that after every 500 picosecond, I have saved its state in a trajectory file, right? So whatever you are visualizing now is a trajectory of my protein. Okay, a trajectory of my protein for how many time? For a time span of 50 nanosecond. This is what you are looking at. ओके, बात समझ में आ रही है आपको कि ये क्या है? यू आर लुकिंग एट अ ट्रेजेक्टरी, एंड दिस ट्रेजेक्टरी इज फॉर हाउ मच टाइम स्पैन? इट इज फॉर 50 nanosecond. Okay, and after every 500 picosecond I have saved a state of my system into a trajectory file. So what you are visualizing now is a trajectory file, right? You can see the motion that is involved in your protein, right? बरोबर है? बात समझ में आ रही है आपको? Are things clear?
Yes, sir.
Okay, so now let me create some really good representation so that things might become more clear. Okay, so now what I will do is that since we, well, let me first we need to create RMSD, right? We need to eventually figure out RMSD between these frames. ओके, सो RMSD कंप्यूट करने के लिए अपने को सबसे पहली चीज क्या करनी पड़ती है? व्हाट इज द फर्स्ट थिंग दैट वी नीड टू डू इज टू अलाइन द प्रोटीन फ्रेम्स. Okay, so that is what I am doing it at the very first step. I am going to extension. Okay, then in analysis we have got RMSD trajectory tool, in which if I hit this align button, I would align all the protein frames upon each other. Okay, so that is done. Okay, that is done. Okay, so now I will create a new representation so that things might become more clear for you. What I will do is that I would first select only protein and create a representation out of it, and I don't want this water molecule, so I would remove this previous representation. Okay, so now only the protein will be visible to you. Okay, this is your protein molecule. Okay, now I would also like to make or I would also like to show you the inhibitor. Okay, so I would select or make a different representation for the inhibitor. Okay, so now since it is only the protein, I would like to show it in new ribbons. Okay, this is your protein in new ribbon, and let me show your ligand molecule by say some CPK so that it becomes more clear. Okay, so now things would be more clear to you. Okay, so now let me decrease the speed of my framing, right? So this is the zeroth frame. That means it is the initial condition of my system, where you can see the diosmine, which is one of the flavonoid that we have tested, that is present in complex with the enzyme called as cyclooxygenase 2, and usually this is the end product of molecular docking, right? तो molecular docking में आपको इस तरीके से प्रोटीन ligand कॉम्प्लेक्स जनरेट करने को आ जाता है, right? So this is how. Okay, then now let me show you the trajectory. Okay, good. So now do you see that the protein has started moving? Okay, you can also see that the ligand also moves inside the active site of protein, right? You can somehow also see that there is motion in your different functional group that are present in your ligand. Okay, is everything visible? Maybe I can increase the speed of these frames. This is okay. Now can you see that this protein is undergoing movement? So is the ligand inside your protein, right? Is this thing visible to everyone?
Yes, sir. Yes.
Okay, so now you can see that you are moving frame by frame, right from starting of the simulation to end of the simulation, right? Okay, so this is how the ligand might orient itself in the active site of your protein. Okay, and performing such MD simulation would give you a kind of idea on what type of interaction it might form by motion as such. Okay, by molecular docking technique you would get only static pictures. So since these molecules are, you know, under always motion, they can orient, the protein can orient its active site amino acid residues. Okay, as well as the ligand can also reorient the functional group that is present in it, so that new interactions can be formed that can be missed out in your molecular docking. बरोबर है? So that is why we perform such MD simulation in order to identify newer interaction if your ligand can make with your protein. Okay, so now you know ki why we have performed this MD simulation, right? And what I have shown you is the trajectory. Okay, now coming to the point where do our RMSD comes into picture. Okay, so now if you want to figure out if my ligand that is shown over here, will my ligand form a stable complex with this protein molecule. Okay, if that is the question that you are asking, then what you can do is that you can plot the RMSD of this system all over the simulation. ओके, बात समझ में आ रही है आपको? अगर आपको ये जानना है कि भाई इफ माय ligand will form a stable, conformationally stable complex with the protein molecule, what you will have to do is you would have to plot the RMSD of this protein ligand system over the entire simulation. Okay, and based on that RMSD plot, then you can determine if your protein ligand system is conformationally stable or not. Okay, so is everything clear till now? यहाँ तक सबको समझ में आ गया है क्या?
Yes, sir.
Okay, so well, now what I will do is that I will tell you ki what exactly happens when you plot the RMSD. Okay, now I will decrease the speed of my simulation to the slowest possible rate as such. Okay, so now what you can do is that you have now you know that we can compare the two states of the protein. You already know that we have compared the structure of hemoglobin with that of myoglobin. Okay, we have got, in that case we have got two different proteins, right? But in this case we have got a single protein, but but it is having some different conformational state. बरोबर है? In previous case, when you tried to compare hemoglobin and myoglobin, what was that case? That was like two different proteins and you were trying to compare the structure of two different proteins, right? बरोबर है? Is everyone with me?
Yes, sir.
Okay, but what is the case over here? The case over here is that it is the same protein that has different conformational states over the given time frame. The protein has not changed, but what has changed? Its conformation has changed, right? Its structural state has changed. बरोबर है? Okay, is
Yes.
it clear? Okay, so now our objective is to figure out if the protein attains the conformational stability when it goes and attach to the ligand. Okay, so now what you can do is that you can superimpose all of the frames that are present over here upon the initial frame. कर सकते हैं अपन. That is what we have done when I went to this extension analysis, right? RMSD trajectory tool, and when I hit this align button, what I have done? I have aligned all the frames that are present in this trajectory to the initial frame. Okay, so now each and every frame that is present in your simulation is aligned to the initial state of your protein. Okay, So now for each frame we can compute RMSD as we have done in the previous case. Okay, बात समझ में आ रही है क्या आपको?
Yes, sir.
For each and every frame we would compute RMSD, and whatever that RMSD is there we would tabulate it. Okay, so we'll say for example when my system got from zeroth that is initial state to, we'll say for example after 500 picosecond it has moved something like this, right? It has moved. Okay, then after again 500 picosecond it has again moved, right? So motion has happened, right? And the state of my protein has changed, right? So whatever the state has changed, we can compute the RMSD between these states then, right? And we can tabulate that RMSD, right? I have already done that thing. Okay, so there is a tool which is present in analysis in this RMSD trajectory tool. You can compute the RMSD by clicking this button. Okay, and all of the RMSD can be saved in a CSV file or comma separated file. You can see over here. Okay, I am opening it up. So here is how what the condition is. We begin with the initial condition, that is at time position 0. Okay, when the system when the simulation reach 500 picosecond, we have saved one of its state of the system, and at this transition the RMSD was 0.98. After again 500 picosecond we saved the another state of the system to the trajectory file, and at that condition the RMSD was 1.077. After again 500 step we again saved the RMSD. In this case the RMSD was 1.23. Okay, and we have done in case of this diosmin. So now we have got how many frames? We have got this 1000 frames, right? So this is your time in picosecond, right? This is over here is the time in picosecond, right? बरोबर है? And we have got this time till, well, say for example all when the simulation ends, it is all around say some 500,000 picosecond, right? 500 nanosecond, sorry, 50 nanosecond makes that much picosecond. Okay, so what we can do now is that we can just plot the graph of the frames with that of your RMSD, and you would get something like this. You can insert, okay, then you can insert a scatter diagram. If you click over here you would get something like this. Okay, so now what do this graph represent? It represents the RMSD that you have plotted all along the simulation of 50 nanosecond. Okay, तो इससे क्या पता चलता है अपने को? इससे ये पता चलता है कि when we started the simulation, right, when we started this simulation, okay, when we started this simulation with initial 10 nanosecond, the RMSD somehow increased. Do you get this thing? There is an initial increase in RMSD, right? For how many picosecond? Sorry, for how many nanosecond? Around 10 nanosecond. And after 10 nanosecond, there is no significant difference, or there is no drastic change in the RMSD of our protein ligand system. Okay, दिख रहा है क्या आपको? So
Yes, sir.
we say that initially there is change in, or there is structural changes that your system is trying to get adjusted with. Okay, and after 10 picosecond there is no significant change in what, in the RMSD throughout the simulation. We say that the system has converged. तो ये कंडीशन को क्या कहते हैं अपन, we say that after this 10 nanosecond your system has converged. converge होना यानी क्या, there is no significant fluctuation in the RMSD, and we say your system has converged. Okay, there are no significant structural changes that happened after 10 picosecond, and hence we say that your protein ligand system has become conformationally stable. Okay, so whatever fluctuation that are happening after 10 nanosecond are only in range of 1 angstrom. दिख रहा है यहाँ पर. Over here we have plotted the RMSD on y-axis and the time span on x-axis. Okay, so after the time span of 10 picosecond, after the time span of 10 nanosecond, there is no any significant change in RMSD. There is no significant fluctuation in RMSD that indicates that your protein has attained conformationally stable state, and it has remained stable throughout the simulation till the end of the simulation, that is 500 picosecond, sorry 500 nanosecond. तो बात समझ में आ रही, are you getting this
Yes,
thing?
yes, sir.
Okay,
Yes, sir.
it is a kind of bit complicated, but this is how you implement the concept of RMSD to determine that in your simulation if your protein ligand has attained conformational stability or not. How would you infer that your protein ligand complex is stable? When there is no significant change in the RMSD throughout the simulation. Okay, when a plateau is reached, okay, do you see that a plateau has reached over here, right? If there is attainment of plateau in the RMSD values in your simulation, then you can say your protein ligand complex has attained conformational stability. Is it clear? Okay,
Yes, sir.
so now what else you can do is that what I have done is that I will show you that this is the simulation of protein and diosmin in complex. What you can do is that you can simulate only protein, which I have already done, and here is the RMSD data shown over here, and I would compare the RMSD of my protein ligand complex with only protein without any ligand. I can insert a scatter diagram over here like this. Okay, now you can see. तो यहाँ पर क्या दिख रहा है आपको? This blue line represent what? This blue line represent the COX without any ligand, and you can see that it is still fluctuating at very higher levels. बरोबर है? While on the other hand, when your COX protein get attached with the inhibitor like diosmin, it gets stabilized. Okay, do you see the graph has remained plateaued? It has converged. The system has converged, and hence we say that when a ligand goes and attaches to the protein, it imparts stability to that protein ligand complex. Right? Whatever you see in blue color, you can see that there is a lot of fluctuation in RMSD. Okay, and this blue colored graph correspond to what? Only the protein without your ligand. Right? So this is how you try to make inferences from your molecular dynamics simulation. So what have you learned from these graphs? That you have learned that when diosmin goes and attach to your COX, the so formed complex is stabilized, and that is not the case when you try to simulate only the COX protein without any inhibitor. right? Is it clear?
Yes, sir. Yes, sir. Yes, sir.
Okay, so now this is one of the application of your RMSD in determining if the molecular dynamics system has converged or not. Okay, is it clear?
Yes, sir.
Okay,
Yes,
so
sir.
I will be sharing all of these files with you guys. Okay, and maybe you can try doing these things, right? This VMD is a freeware. You can download it from University of Illinois website. Okay, so you can visualize this protein system. Okay, I would share this video so that you can follow the steps, right? I will share everything.
Sure.
Huh.
Yes. Yes, sir.
Okay, so this is all I have for you guys today.